Brain rejuvenation breakthrough, how limiting glucose could spark new neuron growth. Nerve fibers, right? From Stanford, California. Could the secret to maintaining a youthful, sharp mind be as simple as watching our sugar intake? A new study from Stanford Medicine suggests that glucose plays a surprising role in the aging brain's ability to produce new neurons. As we age, our brains become less adept to producing new neurons, a process known as neurogenesis. This decline can have far-reaching consequences, contributing to memory loss, reduced cognitive function, and potentially exacerbating neurodegenerative disease like Alzheimer's and Parkinson's. It also hinders recovery from stroke and other brain injuries. However, this new research, led by Anne Brunette, PhD professor of genetics, offers hope by shedding light on why neural stem cells, the precursors to new neurons, become less active with age. Using cutting-edge CRISPR technology, Brunette and her team conducted a comprehensive genetic screen to identify genes that, when inhibited, could reactivate dormant neural cells, stem cells, in aged mice. Among the 300 genes it discovered, one stood out: SIC2A4, and it codes for glucose transporter protein GLUT4. We first found 300 genes that had this ability, which is a lot, Brunette explains. One in particular caught our attention. It was a gene for the glucose transporter known as the GLUT4 protein, suggesting that elevated glucose levels in and around old neural stem cells could be keeping those cells inactive. To validate their findings in living animals, the researchers developed an innovative in vivo screening technique. They injected viruses carrying genetic instructions to knock out specific genes into the subventricular zone of aged mouse brains, an area rich in neural stem cells. After 5 weeks, they examined the olfactory bulb, where newly generated neurons typically migrate. The results, published in the journal Nature magazine, were dramatic. Knocking out the SIC2A4 gene led to more than twofold increase in new neuron production in the olfactory bulbs of old mice. This boost in neurogenesis was accompanied by an increase in both quiescent and activated neural stem cells in the subventricular zone, indicating that the treatment was stimulating the stem cell population itself. Further investigations revealed that neural stem cells from older mice take up about twice as much glucose as those from young mice. This increased glucose uptake appears to push the stem cells into a more dormant state. By knocking out SIC2A4 and reducing glucose influx, the aged stem cells became more likely to activate and produce new neurons. It's allowing us to observe 3 key functions of the neural stem cells. First, we can tell they are proliferating, and second, we can see that they're migrating to the olfactory bulb where they're supposed to be. And third, we can see they are forming new neurons in that site, explains Tyson Roots, PhD, lead author of the study and former postdoctoral scholar in Brunette's lab, in a media release. The glucose transporter connection opens up exciting possibilities for future interventions. Brunette describes it as a hopeful finding, suggesting that it could lead to the development of pharmaceutical or genetic therapies to stimulate new neuron growth in aging or injured brains. Perhaps even more intriguingly, it raises the possibility of simpler behavioral interventions, such as a low-carbohydrate diet that might adjust the amount of glucose taken up by old neural stem cells. While this research marks a significant step forward in our understanding of brain aging and regeneration, it's important to note that the study was conducted in mice, and further research is needed to determine if these findings translate to humans and to explore the long-term effects and potential side effects of manipulating glucose uptake in neural stem cells. Nevertheless, this study provides a promising new direction for addressing age-related cognitive decline and potentially treating neurodegenerative diseases by identifying GLUT4 and other key regulators of neural stem cell aging. Scientists now have promising new targets for developing therapies to rejuvenate the aging brain. The study was conducted in mice, so it remains to be seen if the findings will translate to humans. The researchers focused on subventricular zone, but it's unclear if similar mechanisms apply to other neurogenetic regions like the hippocampus. The long-term effects and potential side effects of SLC2A4 inhibition were not evaluated, and additionally, while the screening approach was powerful, it may have missed some important genes. The study provides strong evidence that elevated glucose uptake contributes to the decline in neural stem cell function during aging, and by identifying GLUT4 as a key regulator of the process, the researchers uncovered a promising new target for potential therapies to enhance neurogenesis in aging brains. The fact that brief glucose starvation could activate good aged stem cells suggests dietary interventions might offer a non-invasive way to boost neurogenesis. However, much more research, of course, is needed to determine if modulating glucose uptake in neural stem cells could safely and effectively enhance cognitive function or treat neurodegenerative diseases in humans. This is from Study Finds by Ann Burnett, Stanford Medicine. Please leave your comments. Thank you for your support. Support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.